Welcome to the Intelligent Polymer Research Institute. This is the headquarters of the ARC Centre of Excellence for Electromaterial Science. Here at ACES, as we're known, we do fundamental work into the discovery and development of new materials. New materials that can be used in energy and in medical bionics. You'll also see through the course of this tour how we're using the most advanced methods of fabrication in order to develop protocols that will enable structures and devices to be created so that we can take those fundamental advances and use them in those important areas of energy and medical bionics. Our first example will explore the discovery and development of graphene. Graphene, that wondrous material that has amazing properties in the nano world, and yet we've been able to extricate those into the microscopic and the macroscopic worlds in order to realize applications. I'd like to introduce you to Sanjeev.
Hello.
Sanjeev has been working with us in developing and discovering new aspects about the chemistry of graphene. What's particularly important is that we modify the chemistries so that we retain all the amazing properties of graphene, the amazing mechanical, electrical properties, and even biological properties, and yet we make it processable. That is, we can take those fundamental, very thin, small sheets of graphene and make them into structures and into devices. Starting with the discovery of graphite, or the mining of graphite from a natural source, Sanjeev, can you tell us something about the importance of that source of graphite?
Yeah, definitely, Gordon. This is the very high purity vein graphite from Sri Lanka, which is mined, and it is 99% pure. So what we are doing is we are starting with this graphite and making the flakes of this material like this one. And after making the flakes, we are intercalating this material. Between the pi stacking, we put the intercalants. And
so just going back to taking the graphite,
yeah.
So we take the lumps of graphite from a mine.
Yeah.
How do we know how pure that graphite is, or what the properties of the graphite?
We have been characterizing this material by CHNS analyzer, how much is the purity of this material, and also checking the impurities by trace metal analysis, how much is the metal impurity, which are very, very important, how much is the impurity content in this one. Once we ensure the quality, then we start processing the material.
And so we take this lump of graphite,
yeah.
And what's the next step? Is it to exfoliate it into different...
First, to make the flakes out of it, and then intercalate those flakes.
So how do we get from here to the flakes?
It is just by manual crushing at this stage. We are controlling the mesh by sieve filtration. And then we do the intercalation of this material.
And that intercalation involves putting other ions between the sheets of graphene in these flakes.
These intercalants need to be of very high boiling nature because they need to stay within the pi space of the graphite. So we have been optimized, like orthophosphoric acid, nitric acid, and sulfuric acids are the main intercalants which we have been using for integrating the graphite.
So we take this lump of graphite, we grind it into flakes, and then those flakes have multiple sheets of graphene, and you're putting ions in between those sheets of graphene.
Right. Yeah, that's
and then
right.
The next step would be?
Then we give a rapid shock, thermal shock of this to this material so that it pops like popcorn. The volume expansion here, this is 2 grams of material. The volume expansion is between 700 to 900 times. So this much of material expands to this much of volume.
Wow.
So this is such a high expansion of this graphite. Once at this stage, now 7 to 9 sheets of graphene are stacked together. The surface area is very high. Now we can very easily chemically oxidize this material.
Right, so now we've got this thermally expanded material and we're going to chemically oxidize it. And does that cause it to break up into the individual sheets
Absolutely.
of graphene?
This is what we have been getting. This is the graphene oxide, which is a very large sheet of graphene oxide, around 50 to 70 micron sheet, which is having a unique property forming the liquid crystalline nature of the material as well.
So people might think, of course, that 70 micron is not particularly large, but it is. I mean, we're talking about a 70 micron sheet. That's a bit bigger than the diameter of a human hair. But remember, we're talking about a material that's 1 or 2 carbon atoms thick, right?
Yeah.
And so the aspect ratio of that is huge.
Absolutely, very, very huge. Yeah.
And that's what causes it to form those ordered structures in solution, or what we would call a liquid crystalline solution,
yes,
right?
That's right.
And then what would you do with that once you've got that form of the dispersion, Sanjeev?
So we have been using this material for a variety of applications, for spinning the fibers and for coating onto the fabric. This is one example. We have coated this material simply onto this fabric, and this fabric is very conducting. As you can see, it is conducting fabric just by simple coating and reducing the fabric.
So you take our dispersion, which is the graphene, and so this fabric is coated simply by dipping into the graphene solution?
Just simply by dipping onto the dilute solution of this material. And after that, it's reduced. Because graphene oxide, there is a lot of defects onto the surface. We restore the aromaticity and it makes the fabric conducting.
And as Sanjeev mentioned, these same dispersions are what we use for fibre spinning, for creating long lengths of individual fibres, as well as for printing, either in 2 dimensions or 3 dimensions. And we're going to be looking at these processes in later parts of the tour. It's really important that all of those chemistries that we've just been talking about are actually scalable. It's one thing to be able to do chemistry on a bench at a milligram scale or a gram scale, but be able to take those same chemistries and process graphene into tens of grams or hundreds of grams and still retain those special properties has other challenges. So Sanjeev, can you tell us a little bit about the challenges we encounter in scaling those processes for graphene?
Yeah, Gordon, on the laboratory scale, when we are doing the oxidation, we are creating defects, we are putting oxygen, and the material after oxidation is insulator. So how many oxygen functionalities we put in? Now we have to, when we are scaling it up, we have to produce the similar quality what we were getting on the laboratory scale, and that's how we will get the final product which we were using before. So it is very important to optimize the parameters here. The parameters what were easy to operate on a laboratory scale, but it might be difficult to operate it here. So what we have been doing is we are optimizing the conditions of the reactions so that we get the similar quality what we were getting on the laboratory. So there are so many parameters which are involved, so we are optimizing one by one.
So I guess that's why it's very important when we're working on the bench that we keep everything as simple as possible, yet try to maximize the properties of the graphene and the processability of it.
Absolutely, you're right, Gordon. So it is important at that stage as well, the critical parameters, of monitoring those parameters which are critical, and then very careful implementation here to see how effectively we can optimize here as well.
And of course, if we're going to explore different methods of fabrication, the quantities that we make on the bench are never going to be enough, even to explore all the different methods of fabrication we'd like to, right? So scalability is really important.
What we have seen in past as well, Gordon, when from batch to batch variation in the laboratory, but once we process larger quantities, there is minimum batch to batch variation, and we can do very effective research using this material from the same batch. And once we optimize this on this scale, after this scale, we can say the technology is scalable. We can go to industrial scale because at this stage we are doing the reactions at 35 liters. And from 35 liters, taking the batch to around 500 liters or 1000 liters is not that difficult once the parameters are optimized.
So in meeting the needs of our experimental program in terms of exploring fabrication, we're also really making this process ready for industry, ready for manufacturing.
Yeah, absolutely right. So once we are confident on that scale, we can definitely go to industrial scale manufacturing.
Great. Well, we're going to go and have a look at some of the different fabrication methods, the fibre spinning and the printing and the making all of these amazing devices for energy and biomedical applications. So thanks to you guys down here for showing us what we do at a very fundamental level in the chemistry of graphene and how we make it scalable.
Yeah,
thanks.
Thank you.
Hi, Ali.
Hey, Gordon.
So Ali's another of our research fellows who's been working on 3D printing and taking all of the advances that we have in the material synthesis and processability and turning them into real structures. Ali's worked on a number of projects. You've been working on the 3D printed ears, the sleep apnea project, and here we're talking about the stents. So what's important about the properties of a stent for particular, for applications?
One of the important part is about the design first. So we have a couple of different design because we have the ability, to code the G code on the machine, and then send it to this machine. This machine is able to print the different structure of with a different property, and plus because we have a different material, we can play with the mechanical property. Let's say, for example, if you add the graphene to the polymer, then we can have the better and robust, mechanical property to compare with the normal polymer.
So using 3D printing, because of the flexibility in what we can fabricate, you can actually customize the stent depending on the particular application. And of course, there can be very complicated shapes of stents that are readily realized with 3D printing.
One of the problem, at the moment for the commercial stent is just they cannot, personalize for the patient. By using the 3D printer, we can make it, personalized according to the scan data of the patient.
Right. And we've been talking about graphene downstairs. Of course, it's an amazing material by itself, but it's often blended with other materials in order to improve the properties of that host structure. And I guess that's what we're doing here, right? You're taking small amounts of graphene and blending that through a polymer that would normally be used to create a three-dimensional stent. Is that right?
Yeah, that's true. Actually, it's just it's allow us to use the graphene and make it printable. Like this structure here, and even a small amount of the polymer we can print the stent like this.
And so that graphene gives it extra mechanical properties?
Definitely, yes.
And I guess if it was needed, could also impart electrical properties into the stent.
That's one of the best benefits of using the graphene for the electro simulation if it's necessary.
Yeah, and all made possible, of course, because of additive fabrication or advances in 3D printing. It's really an exciting time where you can take fundamental discoveries into really practically useful structures almost immediately by you guys working together. Yourself here on the 3D fabrication and Faisal and Sanjeev and you guys on the materials processing.
Yeah, that's true.
Brilliant stuff. Thanks, Al.
No worries.
Hi, Eva.
Hi Gordon, how are you?
I'm very well. So we've just been talking downstairs about the advances in materials like graphene and biomaterials and looking at some of our fabrication aspects. Of course, another important part of our work is the integration of living cells into our printing protocols. Can you tell us something about how we prepare those cells for printing in a facility like this?
So those cells that we have, we culture these cells in this lab. It is a PC2 facility. So we have a risk group rating which allows us to work in an environment that the cells that as a scientist we can be protected from those cells, but the cells can actually be handled in a very safe manner. So we have cells which we obtain from human patients. Cells that are from immortalized cell lines, cells that are from various tissues that we might be interested in expanding and differentiating down certain lineages. So, so that's where these cells are stored in liquid nitrogen. Here we have a couple of examples of where we store our liquid nitrogen to try and ensure that they're maintained at a very cold, in a very cold environment that allows them to be able to be expanded, taken out of the storage and expanded over a long time. We also have an environment where we try to nurture those cells in terms of providing the right growth conditions to expand. So we have incubators that we just go around. So we have here incubators that provides a warm 37-degree environment. A warm 37-degree environment that has air that is good for maintaining a pH. It's an environment that's good for the cells to grow and be conducive to cell growth. So.
Right. And so, do you monitor the health of the cells during these processes,
Yeah.
or do we do that afterwards?
So, quality control of our cells is very important. So, knowing that a cell that is derived from a skin biopsy is exactly that in the first instance is very important. If those cells are then reverted to their pluripotent cell state, a stem cell state, we have to identify that those cells do maintain the ability to be pluripotent. So, yes, absolutely.
And how do you test that?
So there are a number of markers that we use, proteins that are expressed that are reflective of that pluripotent stem cell state. But we also, more simply, take them out of the incubator when they're grown in the tissue culture plates, put them under the microscope.
Oh, let's go and have a look at the
Put them
microscope.
under the microscope. We have Sarah there, she's looking at some of our cells. Put them under the microscope and ensure they actually have the phenotype that we expect to see.
So under the microscope, is it mostly you're looking at the shape of the cell, or are you actually
Exactly.
scanning it to see if there are those markers we just spoke about?
Yeah, so there are different levels of characterization. There's the level of actual just basic morphology, that high nucleus to cytoplasmin ratio that is characteristic of, say, a pluripotent stem cell. But when it comes to looking at those markers, one of the simple methods that we use is immunofluorescent cytochemistry. So we use an antibody that recognises that specific target protein, and if that antibody is linked to a fluorophore, we can see that under
Glows in the dark.
glows in the dark. We
Yeah.
see that under the microscope. We can add a lamp that will mercury lamp that will excite that fluorophore, and we can see it glowing in the dark. Exactly.
Right. So we get the cells in, we store them, we put them in an environment that they like to stay
Yep.
in, and we
We
characterise them
characterise them.
all along the way.
And we grow them. We put them in the incubator to expand, to scale up the number of cells that we need. When we go through the process of bioprinting, we want to create a three-dimensional tissue, not a few cells in there. We want to actually have a high cell to biomaterial mass. And so what we want to do is scale up the number of cells that we have to be able to encapsulate into the biomaterial, which we will do next door. But that process of encapsulation, of working with the cells, it's very important that we do it in a safe environment such as we do in the biological safety cabinets that we have over there.
So are there other characterisation tools for the cells that you use in here, or should we go have a look at the bioplotter?
We can. It's a process of going back and forth.
Right.
So whilst we scale up the cells here in the first instance, we go to use a bioplotter and we print those cells in a structural form that we'd like to have, but then we bring it back into this lab and we continue the culture and growth. The characterization also extends to, say, a functional analysis of, say, if you're looking at nervous tissue or neural tissue, you'd be looking at multi-electrode array, so look at those electrophysiological responses.
Right.
Or you'd perhaps be using confocal microscopy to look at calcium flux, how those cells network and show coordinated flux.
Okay, well, let's go and see how we build all those cells into the actual 3D printed structure. So having prepared the cells in this sort of environment, we go take them to the bioplotter, and then create that structure.
Yeah.
But of course, we'd want to bring that structure back into this environment to
Yes.
interrogate the cells again,
Yes.
see if they're still living,
Yes.
if they're proliferating,
Yeah.
and maybe even turning into the types of cells that we want them to. Is that
Yeah.
correct?
That's correct. Another really very important point is actually demonstrating that the biomaterial that we're actually creating, that nested material, that encapsulated material, is supportive to just basic cell viability. So we have cytocompatibility testing that we also do. That's another aspect.
So if you were to bring the cells here, what would you actually do? You'd load them into this 3D bioplotter?
So we
Maybe
bring-
if you could just point out how we do that?
Certainly. So what we have is a 3D bioplotter here. There are cartridges that we load our biomaterial that has the cells encapsulated, or just the biomaterial alone. We don't need to have the cells necessarily at this stage encapsulated. We bring that already prepared in a cartridge. That cartridge, almost like a syringe cartridge, that cartridge is loaded onto these cartridge holders on the right-hand side. They are temperature controlled, so it's conducive to bioprinting at particular temperatures that allows the form of the filaments to be preserved.
So the cell, at this stage, the cells would be added to the BioInk formulation, is that correct?
There are two ways we can approach this. We can actually allow the cells to be encapsulated into biomaterial, or we can actually seed afterwards. So this is the part where there's an option of one or the other. Having said that, the biomaterial that we bring, that we have prepared under sterile aseptic conditions in the tissue cabinets we have over there, that is then brought here. And yet again, we are in a aseptic environment. So we have a BSE class 2B cabinet that we introduce our cells or our biomaterial with our cells into this environment.
Right. And I know recently we've been successful in printing structures containing induced pluripotent stem cells. That's an amazing advance, isn't it, for both fundamental studies but also potentially for clinical applications?
Absolutely. Pluripotent stem cells have the ability to differentiate down all the different lineages of the body. So we can allow the cells, it's what's quite remarkable is that the cells, which are very difficult to handle in native culture, we can actually incorporate that into the BioInk, which allows the viability of the cells to remain viable and to promote the incorporation of the cells into the material. So yes, we can print the material under with the bioplotter into a defined structure, a three-dimensional structure, and then that material, once we take it back into the PC2 lab, we can culture in a media with growth hormones and other supportive media, additions to the media that allow the cells to differentiate down different lineages.
Right.
So it's quite exciting to be able to create these very high density structures that the cells are able to form a specific 3D tissue.
Yeah, and so that ability to create those structures gives us fundamental insights into how things behave in 3 dimensions.
Yes.
And obviously what we're discovering is it's very different from 2 dimensions.
Yes.
And so
we're not a flat body.
Yeah.
We're not a flat surface. Our cells in the native environment in our bodies interact with other cells in that 3-dimensional space, interact with the extracellular matrix, and that biomaterial provides that extracellular matrix that the cells can have support to grow further and to really integrate into the 3-dimensional tissue.
So it must really bring into question the validity of pursuing studies in 2 dimensions in terms of conventional cell culture.
Yeah, it's a growing. It's been a growing concept that's 2D is great for fundamental initial studies, but taking these studies, from whether it's the cancer cell biology or basic tissue biology, taking cells from a 2D matrix, a 2D monoculture to a 3D matrix is essential to understand tissue biology.
Great. Well, thanks for giving us that insight into the preparation of the cells for 3D bioprinting and then how they continue to be interrogated after 3D bioprinting in order to understand how they develop in 3D and how we can manipulate their development in 3 dimensions. Great stuff.
Thanks,
Thank you.
Gordon. Thanks for your
you.
interest.
Thank you. Hi, Javid.
Hi, how are you?
Oh, very well, very well. So we've just been talking to the guys at the early part of our lab tour about, the graphene, how we get the graphene from the graphite and use all that amazing chemistry, to get those stable dispersions and then scale that process. But of course, you're the one who's weaving all the magic here with those dispersions and turning it into real fibres that we can actually handle. So how does this process work? You know, you get the dispersion. What do you do with it next?
Okay, sure. We starting from here, we make a spinning solution like the honey, and we inject it through the pump. This is the lactic pump and the solution, and it's go through the tiny spinneret. This is like that, and inject the solution to non-solvent system. This means the solution converted to the solid material.
So by non-solvent system, so we get this solution or dispersion as it goes into the bath. It basically precipitates, right?
That's
it
true.
comes
Yeah,
together.
sure. Yeah. And just in the beginning, this is like the hydrogel shape, and after that, it become dry and become solid state. This is completely. This is like that, or like that.
Right.
You can use this one as a strong fiber. It's graphene fiber based on the Wettesband structure.
Right. So it comes out of the bath like that gel state that you just described to us. So,
Yes.
So that comes out of the bath, then gets collected on the rollers. And
this collected through here, and the next step is just improve the mechanical property by the number of the coagulation bath. We can use a stretch it, we can wash it, we can add the other additive you want. For example, any biomaterial you want to coat in the surface of the fiber or improve the electrical or mechanical property, we can do through this step. And we can, of course, we can control the temperature, we can control the tension during the spinning process. It completely optimizing the machine. And the end of the process, we can dry the fiber through the heater. There is a heater here, and we can collect the fiber. End of the process like that. We can get a spool of the fiber, depend on the material. For example, this is a man-made fiber, polyester. We can produce or graphene fiber through the depend on the application. We can get the single fiber or multi-fiber collected.
so it's really quite a simple, scalable process, but obviously each one of those steps needs to be very carefully controlled to get the properties that you want in the final fiber, right?
Yeah, that's true. Yeah, it looks very simple. All this technology for fabricating material, but it's still powerful, and we can tuning every property you want for future application. If you look at the using for variable technology, of course, you need a superior mechanical property for the fiber. Sure, we have to enhance the property here, and later we can use for the variable technology. If you're looking for the implant electrode, of course, the other property we can improve here, like the electrical conductivity and biological activity through this process.
And as well as the graphene fibers, I know recently we've been doing work where we're actually spinning long lengths of biomaterial fibers, right?
Yeah.
Where those biomaterials might have a different function from the graphene for particular applications.
Sure, there is in particular application we use the Olvan. This is the new material, and we can of course convert it to 3D structure through our facility like knitting, braiding, or other type of the 3D fabrication currently we have.
So welcome to Tricep. This is a new facility that we've created in order to take the fundamental advances in material science and engineering into a manufacturable prototype or a commercial product. You've seen all of the fundamental work that we've done, for example, with graphene and other biomaterials, what we've been doing in terms of processing cells for 3D bioprinting. And you've also heard about some of the practical applications where we can use that fundamental knowledge. Here we focus on those practical applications, and we focus on developing protocols for the bioinks that make them ready for manufacture. We focus on developing the protocols for the customized printers that make them ready for manufacture. This is an exciting new development, and we're going to have a look at what we're doing in those areas of bioinks and bioprinting, and how we're addressing some really critical medical challenges by creating that commercial opportunity. So we'll go and have a look at Tricep. This is a great facility incorporating all of the bioprinties, development of new bioprinters using conventional 3D printing, but also, as you won't find in a conventional university research environment, taking all of those processes and making them ready for manufacture by implementing a quality management system that allows us to realise real commercial opportunities. I'm going to have a chat with Alex about what he's been doing with BioInks. Alex works here at Tricep. He's an employee of ANF, the Australian National Fabrication Facility. So, Alex, you get the fundamental formulation of the BioInk from our colleagues across the road at IPRI and ESSES. And so what challenges then do you have in terms of really scaling that up and turning it into a reliable product?
So everything with upscaling becomes a lot different. Every parameter of these experiments and the results that we get out have to change. So what we have to do is really explore every single parameter in these experiments piece by piece, so that when we are making larger batches of these BioInks, they'll be, as you said, reliable and the same. So there are a lot of challenges, but it's a slow working process.
And so I guess we also really need to go right back to the source of the materials, don't we?
Yeah.
I mean, for some of the research work that we're doing, of course, getting small amounts of material in order to do those small scale experiments is relatively easy to achieve. But here you really need to know what's the primary source of those materials in order to ensure quality control. Is that correct?
Yeah, that's correct. So we have a really nice network of collaborations around UOW and Tricep. The one that comes to mind is a lady called Pia Winberg. She is our main collaborator in producing us with a type of seaweed called ulvan. And we here are making BioInks from ulvan. And it's a part of that nice collaboration that also helps us know that we have a high standard. So before we start making any large scale production of the BioInks, we have to undergo a suite of characterization to know that they're high purity and then, a very good product for us to work with.
So you would characterize that material on arrival to ensure its quality, and then you would start to do whatever reactions you need to do to turn it into the BioInk. Could we have a look at one of the reactors you would typically use?
So we start off at these smaller scales. This is a good example of a reaction that we would particularly do over at AIM. And so taking the results that we would make from this, we would hope to achieve similar results in the large scale reactors. So I'll take you over to the...
So what sort of quantity of material would you be generating from a reaction of this scale?
So in this scale, I would say you could obtain 20 to 50 grams is the scale.
Okay, so that's still quite large scale compared to what they would do in the research facility, right?
Yeah, definitely.
So it's one step up.
Yeah, research is about 1 gram. We're working towards 50 grams here.
Right.
And now I'll take you to the reactors. These reactors are state of the art. So these reactors, this is a 10-litre reactor. This is capable of generating quantities of up to 500 grams. All of this has been controlled by a thermoregulation unit, which is plumbed through the external walls. We can control the temperature extremely sensitively. We can collect our material from the bottom, which means everything remains extremely consistent when comparing it from the small scale to this larger scale.
Yeah.
And over here, we go up a little bit further, and this is our 20-litre reactor. And this 20-litre reactor is capable of producing kilogram batches. Kilogram batches is in my opinion, the big leap into an industrial process.
Right. And so Sanjeev was highlighting to us when we were talking about the scalability of graphene processing, the importance of simplicity. So what are your thoughts on that?
I agree. This is as simple as it can get without reinventing the wheel. This has a simple heater and a jacket to keep it warm with just a big motor to stir it. It's as simple as it can get.
Yeah, and I guess it's important that the actual chemistries used are simple and is the minimum number of steps possible. Right?
Exactly. So in the work that we've done here, we've managed to reduce steps significantly from the lab scale. So from the 1 or 2 grams, we're able to reduce those 10 steps into 2 or 3. And that's a big part of process chemistry.
Yeah, and that's a huge step forward, getting down to 2 or 3 steps which you need to control accurately. And I guess once you have that material, then you've characterized what went in, and then I guess you need to characterise what comes out.
And
right?
so the first step in doing that process is to purify it. So on a kilogram scale, this is our new piece of equipment that we have recently customized. This is a cross-flow dialysis purification system.
Wow.
This system is capable of running through about 140 litres of our reaction mixture. It'll pass through a membrane that sits in here, which works like via dialysis, and it will throw away any side products and impurities that we don't want, and leave us with the pure material in this batch.
And so compared to processes that would have been used before, what sort of time saving are we talking about here using the cross?
So on the small scale, we would be doing dialysis in dialysis bags, which could take up to 3 to 4 days. This allows us to generate, and that was again limited to 1 gram. This allows us to generate kilogram batches in 2 days.
Wow. We're going to talk to Steve about some of the aspects of the bioprinters. We must be able to commercialise and complete the whole package in order to address the clinical challenges that are in front of us. Hi, Steve.
Good afternoon.
So Steve is heading up our additive fabrication capabilities here at Tricep, and he's going to introduce us to some of our capabilities. It's a capability that we like to call printing printers. So tell us about that, Steve.
So we've been very fortunate. We have a range of additive fabrication tools, and we've recently been able to expand on that set of tools. This is just one of the new printers we have. It's an Omaki 3D printer, allowing us to print components in full color representations. And that's now allowing us to give much higher level of details to our collaborators and our commercial partners to get real realizations of the parts that they want to be able to produce. These systems are then enabling us to print new components for new printing systems, and we'll be able to run you through a couple of those systems right now.
Of course, all of these projects we're going to look at are driven by real clinical need. I mean, we're very fortunate, aren't we, to be able to work with clinicians all around the country who are really defining the challenge with us, defining the plan and implementing the plan, and then helping us in terms of translation.
That's the real driver behind what we're doing, and it's giving us great encouragement to work outside of just doing research for the sake of research and being able to treat real clinical needs. And this is one such example. We're referring to this as the iFix pen. This is a device that allows us to deliver media or deliver two materials straight to a corneal ulceration in the eye, allowing us to treat that corneal ulceration, and in either the domestic environment or an overseas environment to be able to limit the chance of infection and ingress into the eye.
So the bio-ink is obviously an important part of this, and that ink has been designed to be compatible with the eye, to be transparent and to facilitate the healing process. And then I guess that the hardware or the iFix pen was designed around the requirements to deliver that bio-ink.
That's exactly correct. And the reason why this is, we always see these delivery mechanisms as tools. They're allowing us to take that fundamental material, the critical therapeutic material, and deposit it in the structure that's required. And this is one primary example where we're taking two materials, mixing them in a custom tip, allowing those two materials to cross-link in gel and be able to adhere onto the eye surface.
And it's been great to work with the clinicians as well, just on practical aspects of the ergonomics, how you hold it, how you deliver it.
And I'm probably holding it incorrectly as we look at it. I believe it's to be held in that manner. Right. But this allows us to refine that. And with the set of tools that we have here, it's very easy to go into an iterative process to refine the structure, refine the ergonomics of the structure so that they have better use of it in practice.
And I guess another thing about Tricep is that we'll go that step further. Not only will we develop the prototype, but we will make sure that that prototype is ready for manufacture by working with commercial partners to do that.
Again, that's a huge advantage of this facility, and building on our existing commercial relationships through linkage projects that we've had in operation for many years, and defining what really can be manufactured at scale. Yeah. So we have, obviously, as we're designing components, we have to keep in mind design for manufacture rules and larger scale injection molding rules, which would be the typical manufacturing processes that we use. but also being able to take our customized prototypes and put them into more complex stages. So the example case of a handheld device is ideal for a practical use in a clinical environment, but for us to characterize that device, to have fine control of the device as we're going through our experimental protocols, it's ideal to be able to transfer and mount the system into a 3-axis stage or even a multi-axis stage to allow us to have fine control over the scaffolding structures that we produce, to then be able to determine the repeatability and the consistency of the printing system that we've developed.
Yeah, brilliant. Well, that's brilliant. I mean, so there must be a whole suite of these that we're involved in developing now.
There is now. So the iFix pen is one of the earlier, simpler examples where we've got two materials that are being extruded simultaneously and mixing at the tip. Another example case, which has a slightly higher level of complexity, is the Excelda pen. And the Excelda pen...
This used to be the BioPen?
That is correct.
Right. Excelda sounds much better.
It does. It's undergoing a rebranding. So with the Excelda pen, we have a slightly more complex extrusion geometry. This is a coaxial extrusion geometry, where we would have an inner core and an outer sheeting of material, allowing us to maintain cells within the inner core and then have a protective outer sheeting during the actual extrusion process. Obviously, this tip is then a bit more complex, and through some of the technology that we have here on site, we've been able to develop a robust, refined structure with a complex internal geometry that's been produced out of titanium alloy. So that's easily cleanable, easily sterilizable, and reusable in our preclinical trials.
So how did you make that?
So the design was done in-house. We optimized the internal fluidic pathway through a CFD analysis, and we were then able to produce it using metal additive fabrication. So the actual process is referred to as selective laser melting, allowing us to produce features with a minimum wall thickness in the order of 200 microns.
So it's printed on a metal printer, a 3D metal printer.
That's correct. So a 3D metal printer, we're very fortunate to have two of these systems available to us. The first is would have a cylindrical build volume, a 70 millimeter diameter tray with a maximum build height of 74 millimeters. The newest, most recent system that we've added to the facility is a Concept Laser 200R. That allows us to have a print volume of 100 millimeters by 100 millimeters by 100 millimeters. So a liter of volume that we can print within. We can print in a range of different metals. Stainless steel and titanium will be two of the most common materials, allowing us to produce high resolution, low surface roughness components that are ideal for customized prototypes.
So some of these structures must be really difficult to make at all using conventional manufacturing approaches.
Well, that's absolutely correct as well. So what you can do is, if you actually look at the supporting structure, and this is sacrificial supporting structure, we're able to make these intricate geometries, open porous lattices that in our mind are ideal for complex electrode structures, or indeed the production of complex fluidic geometries.
Yeah.
Okay.
But that's amazing. I mean, so you really are printing printers.
Well, we're moving on so that we, instead of having our handheld devices and integrating those into commercial type platforms, we're actually developing our own customized platform that's allow us to have additional functionality to incorporate additional extruder or deposition techniques simultaneously, as well as cross-linking methods. So in this example case, the 3D Ed, we're able to have two materials being simultaneously coaxially extruded with temperature management on both of those materials, and also the feature or the capability to be able to cross-link those materials as they're being deposited.
And this educational printer is an important part of our online teaching program that we're putting together. As you know, we have a graduate certificate in biofabrication, which is available online now. And that is actually the coursework or that's necessary to move on to the Masters in Biofabrication. And it's exposure to all of these types of printers that really give high-level, state-of-the-art training in 3D printing, isn't it?
That's right. And within this small package, our prospective young engineers and bioengineers have a range of tools, a range of capabilities for them to learn. So from the fundamentals of the 3D positioning systems right through to the development of the manual or mechanical extrusion mechanisms, temperature management, to allow us to see what happens to our different materials under different temperature conditions, but also under different printing conditions or extrusion diameters.
And of course, building the printers is in itself a great dimension to the training, isn't it? We had a number of summer scholars working here with the team at TriCept, who actually were the first, I guess, to build the 3D Ed, and we're learning from that experience and how we can use that building of printers as a training aspect.
Yeah, and it's been a training session for us as well. Those students were able to identify the benefits for them in what they were able to grasp from our initial developments, and what they were able to contribute towards it. So to how we could refine the design to improve our design of manufacture and design of assembly.
Great. So thanks for giving us that amazing insight into how we're using conventional 3D printers to create a range of customized printers, which is helping us to address important research challenges in the medical sphere, but also helping us to build new tools for training the next generation of biofabricators. Thanks, Steve. Brilliant. Thank you. Hi, Zhao.
Hi, Golden.
How are you doing?
Yeah, I'm great.
Good, good. So I've just been talking to Steve about all of this range of customized printers that we're making for our clinical collaborators. What's the clinical project that you're involved in?
This one is for the pancreas islet transplantation project. And this is the printer we developed for this project. We call it 3D PICT. It's the pancreas islet cell transplantation device. And in this device, we contain the 3-dimensional printer with the core and shell structure. And this is some sample print in the core shell structure.
So the core and the shell, in this case, Steve was telling us about putting the cells in the middle of the core. What's the idea here? I remember that we need to integrate 3 different types of cells for the islet cell transplantation. So how would those 3 types of cells be integrated?
Yes, we encapsulate the pancreas islet cells in the core structure, which can provide more immunoprotection for the islet. And we can put the supporting cells such like endocellular progenitor cells and in the shell structure, and these cells will promote the vascularization process of the whole implant construct.
So
in the meantime, we also have this droplet device. They can drop the cells into the printed construct. So we have the potential to deliver 3 types of cells together into one construct, such as Treg cells, and these cells can protect the islet from the immunoprotection from the patient itself.
So the islet cells, they're obviously the key cell that the clinician wants to transplant into the patient. So they come from a donor, and they are going to respond to glucose and release insulin. And then you mentioned the endothelial progenitor cells, so they will help with vascularization, is that correct?
Yes. Yeah.
And then you mentioned T regulatory cells. Now,
Yes.
these T regulatory cells, as I understand it, they help to regulate the immune response, is that correct?
Yes.
Right. So this printer then can deliver all 3 types of those cells discretely in a 3D structure.
Yes.
Can it also spatially distribute them where you want them in order to optimize performance?
Yes, correctly. We can control the position of each type of cells. And for example, we can put the Treg cells in the surface, in the outer layer of the construct, and protect the islet cells inside the construct. And in the meantime, the EPC cells will go through whole construct and promote the vascularization of whole construct.
Great. And I know that this project is carried out very much in association with Professor Toby Coates' group in Adelaide. So how does that work? I mean, do they now have a printer in their laboratory?
We deliver a custom-made, is nearly identical to this one, to their lab, and they also work hardly on that device and try to print the construct for animal work. We already implant some construct in mice, then we are targeted to the large animal samples for such as pig. Yeah.
Right. So it must be exciting to be involved in a project where there's very fundamental advances in materials, fundamental advances in how we understand the cells to work in combination with those materials, and then actually delivering those ink formulations in the hardware into a clinical environment where we can envisage translation of this technology to help individuals. That must be exciting.
Yeah, we are really excited to get involved in this project. We are not using our knowledge in the material and the biology and engineer and to develop the device and formulate the bioink formulation, and which can be used in the clinical and have potential to treat type 1 diabetes.
Brilliant. That's great. Thanks, yeah. Thanks
you.
for providing those insights for us. Thank you. Well, we have one last customised printer to look at. So I'm going to introduce you now to Johnson. Hey, Johnson.
Hello.
How's it going?
Good. How are you?
And our great friend 3D Alec.
Yes, this is the 3D.
What's Alec all about?
So this is a printer that we built for the clinical collaborator at Sydney RPA Hospital. So all of the components were built in-house, and this printer is able to print 3 different materials, each having their individual printing speed and conditions. So we sort of tailor them to be able to print different materials that require different printing parameters.
So, like all of our other customised printer projects, of course, driven by a real clinical need, and usually by, well, always actually by a passionate clinical collaborator. So, tell us a little bit about our passionate clinical collaborator on this one.
So this printer was designed for the project where we regenerate the cartilage of the ear. So it's in patients with what's called microtia, and that's in the normal development of the normal ear. So what happens is that the ears, when the child is through the growing stage, they will have a deformed ear where they only have a little bit what's called a peanut shape ear. So it's different from a normal size ear, and the current techniques to treat that requires a lot of surgical process and leads to a lot of complications. So the passionate clinical collaborator came to us looking for a bioprinting solution. That's where we came up with this idea to print the entire ear out of biomaterials.
Right.
Yeah.
And so, and of course, that passionate clinical collaborator is Pal Mukherjee from the Royal Prince Alfred Hospital. So this is obviously quite complex, right? Because you need I'm guessing, a material that's going to retain structure, mechanical integrity, and another material for growing cells, and in your case, cartilage. So I guess that's why we have this multi-head printer that's been necessary for this project.
Yes, as you can imagine, if we were to just print an entire ear scaffold out of a gel-based material, it's not going to hold shape. And the ear is all about the aesthetics. So that's why we needed materials that give it the structural support, materials that give it the cell support, and then obviously a sacrificial material which is required for all 3D printing applications. That's why we have this complicated printer where we have 3 heads, one that cools down, 2 of them that cools down, and 1 of them that heats up the material to allow us to do melt extruding.
And so we've developed this with our clinical collaborator who's helped us to articulate the challenge and then develop the plan and put that plan in place. Where are we now in terms of transferring this into the clinical research environment?
So as you can see with some of these scaffolds, they come with different pore sizes, different orientations, and each of these different patterns correspond to a specific stiffness and flexibility, just like the normal ear, what we experience in the normal ear. So this is sort of the first phase of a project where we try to represent each of these regions by changing the parameters of the printing process. Then the second stage is then to incorporate cells in there and induce chondrogenesis, and which is what we're trying to do right now, inducing chondrogenesis in each of these different patterns and hopefully each of these regions because of the different stiffnesses and mechanical properties they would produce allow these chondrocytes to excrete out different amounts of matrices.
So obviously a very multidisciplinary project, bringing together material scientists like we've met today, and process engineers as we've met today, and mechatronic engineers and biologists and biomaterial scientists. I'd imagine that projects like this are both challenging but extremely rewarding. More challenging or more rewarding?
I would say a bit of both. It's definitely very challenging as the ear isn't one uniform stiffness as we can see. It's a whole range, and then we have to make sure select inks that are not only printable but also compatible with the cells, and that's sort of the challenging bit. The rewarding bit is once we can introduce this new concept, then we sort of solve a lot of the issues that we have currently in surgery where they have donor side mobility. And so this is a lot easier in terms of the printing process.
Well, we've had an exciting journey around our labs today, starting with the fundamental advances in material science and how we're using chemistries to retain all of those amazing properties we discover sometimes in the nano world of materials, and bring those through into processing structures like the sutrode and making devices like for the ear and the cartilage regeneration, and blending together all of the skills necessary to do that. And of course, working very closely with all of our collaborators who are real end users. And here at Tricep, being able to complete that pipeline to take everything to that manufacturing level, you know, the bioink that can be manufactured, the printer that can be manufactured, and all of those solutions delivered to our end users. It must be a very interesting experience to be involved in here at Tricep.
It is. Over here, you can actually put your projects, the research project, into practicality, into a clinical scenario, and that's what's exciting.
That's what we're here for.
Yes.
Cheers.
See ya.
Well, we hope you enjoyed your tour of IPRI, the headquarters of ACES, and the introduction to Tricep, where we're making things translatable so that we can solve real applications using fundamental advances in material science and engineering. If you have any further questions, please don't hesitate to contact us or hang on and ask us now. Thank you.